Hi, today I would like to talk about this presentation named Matrix metalloproteinase 3 regulates angiotensin 2, induced myocardial fibrosis cell viability. What does matrix metalloproteinase means? It's actually an interesting question. I want to learn about it, so let's learn together. In many biological procedures, including embryogenesis, restructuring of tissue, wound healing, and angiogenesis in pathological conditions like arthritis and cancer matrix, metalloproteinases play an important part in this process. 24 genes in humans currently encode different groups of matrix metalloproteinase enzymes have been identified. Their review discusses the specified structure, function, and regulation of the enzyme activities by tissue inhibitors of members of the matrix metalloproteinase family. So, I would like to talk about this slide. Heart failure has become a major public health issue affecting 23 million people worldwide, and heart failure constantly increases due to life expectancy over time. There are 24 different genes to encode the NPV family. These genes are coded by 5 different gene regions. These are the signal is peptide, propeptide, catalytic site, the hook region, and the homopexin-like region. Of the signal peptide, propeptide, and catalytic domain are found in all NPP3s. All in NPPs. Gene regions with N-terminal ends, and it starts with a C-terminal 9. The propeptide at the N-terminal end contains 80 amino acids. With the proteolytic removal of this region, NPPs become active. Cysteine residues in the propeptide region plays a critical role in keeping NPPs inactive. The cysteine residues binds with the zinc in the catalytic area to prevent the activation of pro-NPPs with the sulfhydryl group it contains. This is called cysteine switch 10. The signal peptide controls the release from the cell. Catalytic zone. It consists of 180 amino acids. The histidine residues it contains from 3 separate binding sites from the zinc ion. The cysteine residues in the propeptide region inactive NPPs is 4 binding site for the zinc ion. The catalytic zone is composed of 3 alpha helices and a twisted 5-strand beta sheet. So, let's go to other slide. Material and methods. Actually, this is one of the most interesting parts of this slide. As you may see, there are 7 material and methods for it. For example, cell culture, ANG2 concentration screening, cell transfectation, cell viability assay, cell apoptosis assay, cell migration assay. So, let's begin with the most useful one, cell transfectation. Other NPP genes such as growth factors, cytokines, tumor formation, hormones, chemical agents, or physical stress are expressed selectively in different cells. For example, exposure to ultraviolet B causes an increase in the gene expression of NPP3 and NPP9 in dermis. Again, matrix cell intervention can also induce NPP expression for the mediation of ingredients. Cell apoptosis assay. What is it? It is actually in order to measure H9C2 cells. Apoptosis according to the protocol of the manufacturer, annexin apoptosis detection kit was used. H9C2 cells were sworn into a 24-well platform and cultivated at 47 degrees for 24 hours. For 5 minutes, the H9C2 cells were harvested and centrifuged, and cells were subsequently double stained at 20% for 20 degrees for 20 minutes with the annexin of 5 milli and the P1 of 10 milli in the darkness. A fax AC caliber flow cytometer and apoptosis rate were used for the evaluation of the cell apoptosis. So let's go to the next slide. Next slide is all about cell migration assay. What is cell migration assay means? Let's learn together. Transfer chambers were used to evaluate migration of cells from H9C2. The upper chamber was sealed with complete medium. The medium was added to the lower chamber with 10% of FPS. After incubating at 37 degrees, the cells were fixed for 10 minutes at room with 4% of paraformaldehyde and stained at the room temperature for 10 minutes at 0.5 violet crystal. Thin cells with a light microscope were visualized. What cell culture means? Cell culture means that H9C2 red myocardial cells were cultured in DMM, supplemented with 10% of FPS. And also it includes penicillin and 100 mg of streptomycin at 47 degrees with 5% of carbon dioxide. So effects on the MP3 on cell migration. What is it? As you may see from the picture, the transfer was conducted to assess the cell migration impacts of the MP3. The number of migratory cells in the MP group decreased in comparison to the control group. The number of migratory cells between the groups MP and MP plus CNA in C was not significant. However, MP plus liver carnitine and MP plus C-arena MPP3 groups, the number of migration cells was significantly lower than MP. The results show that MP3 knockdown inhibited additional cell migration in ANG2 treated H9C2 cells. Effects of MMP3 on cell apoptosis. As you may see, I included a picture that you can understand it better. So, what is it? You're going to ask. Let's learn together. ANG2's effects were detected by cytometry on H9C2 cell apoptosis. Cell apoptosis 0.43% for control, MV and MV plus C-arena in C group respectively. The results showed a significant increase in cell apoptosis rate in the MV group compared to that in the control group. While the rate of cell apoptosis between MV and MV plus C-arena and C group has not been significantly different, the rate of apoptosis in MV plus liver carnitine and MV plus C-arena in MPP4 groups was substantially increased compared with MF group. The results showed therefore that ANG2 increased apoptosis of H9C2 cells compared to control cells and further reinforced ANG2's induced apoptosis of H9C2 cells. Results. In addition to physiological events such as the development process, the MPP3 enzyme family and its inhibitors have been shown to play a role in many diseases, such as connective tissue disease, lung disease, cardiac disease, skin disease, and some metabolic disease, is increasing in importance. Although MPP enzyme family and inhibitors can be used as potential agents in the treatment of this disease, it is true that in the future MPP genes will gain importance in personal